2 miles down, in water cold enough to stop a heart and pressure that could crush a submarine, a single blue-green light begins to appear, swaying slowly in the absolute dark. That light belongs to an anglerfish. Because of how it uses that beacon to lure prey, we assume the fish is producing it, but it actually has no biological mechanism to create bioluminescence on its own. The glowing tip of its fishing rod, called the esca, is a specialized physical couch. Inside that pouch, the fish cultivates colonies of luminous bacteria. The fish doesn't shine, the bacteria do. The most famous glowing predator in the ocean is essentially a wandering host carrying a jar of living microbes on its face. Since the bacteria are essential for survival, the logical assumption is that they are inherited, passed directly down from a mother anglerfish to her offspring. This animated diagram maps the family trees of both species side by side. On the left, we track the anglerfish lineages. On the right, the bacteria. If they evolved together and were passed from mother to baby, these lines would march down the generations in perfect step. Instead, they cross and mismatch entirely. There is zero evolutionary alignment. This tells us every baby anglerfish hatches into the abyss with a dark, completely empty lantern. At some point, it must open that pouch to the seawater and recruit specific bacteria, Candidatus Enterovibrio, directly from the open ocean. Nature forces this creature to start from zero every time. This iconic partnership has to be built from scratch in the wild, generation after generation. Out in the open seawater, before they are caught, these bacteria drift alone, and when they are alone, they stay completely dark to conserve energy. Creating that glow relies on pure chemistry. A molecule called luciferin reacts with an enzyme called luciferase. When exposed to oxygen, the luciferin burns in a controlled way and throws off a single particle of light, a photon. But glowing alone in the dark ocean is a waste of resources, so the bacteria use chemical sensors to measure how crowded they are. They wait until they are densely packed inside an anglerfish's pouch before they all switch on at once. The intense brightness of the lantern is dictated by density. It requires 1 billion separate cells, sensing their neighbors, and making a coordinated chemical decision to shine. This scale compares the energetic expense of cold light. A tiny block is standard cellular energy, 30 joules per mole. The massive block is a single blue photon, 255 joules. To fund this flash, bacteria snap a peroxide bond, releasing stored power. Evolution rarely invents something this expensive from scratch. Scientists found that the glowing molecule, luciferin, originally evolved as a chemical shield to mop up dangerous oxygen. The glow was a byproduct that nature eventually repurposed into a signal. Because producing light costs so much energy, the free-floating bacteria cannot afford to do it often on their own. To keep the beacon shining, the anglerfish pumps pure, high-energy glucose directly into the lantern. It is a strict, localized economy running 2 miles below the surface. The fish feeds the bacteria sugar and buys their light in return. Since these bacteria are caught fresh from the open ocean every generation, they are technically free agents. Their internal biology should look like any other independent microbe. This diagram shows a typical circular genetic sequence. But when scientists read the DNA of the bacteria living inside the anglerfish, they found an anomaly. The bacteria permanently lost 50% of their genome, discarding the instructions needed to make amino acids and feed themselves. In biology, massive gene loss is the classic signature of an ancient, trapped parasite that relies entirely on a host to survive. Yet these bacteria still possess swimming tails and environmental sensors, and they are recruited fresh from the water. The facts contradict each other. We have independent, free-floating microbes that are actively decaying exactly like long-term inescapable prisoners. Biologists call this a third symbiosis. It is a newly discovered category of life sitting entirely in the gray zone, somewhere between independent organisms and permanently locked-in parasites. The anglerfish lineage is around 100 million years old, but the bacteria inside them are still actively shedding DNA today. This is an unfinished evolutionary process happening right now. When you look at an anglerfish moving through the abyss, you are watching a live, mid-sentence evolutionary experiment. The darkest, most hostile environment on Earth is illuminated by a biological compromise. Two entirely different species found each other in the void, and they are slowly fusing into one.